Hi everyone, today let's continue the chapter 3 of the Cellular and Molecular Immunology from Abbas, the book that I'm using in the series of lectures. And last lecture we were discussing or introducing the trafficking of the leukocytes, the cells of the immune system, right? And I superficially introduced that for the communication of the tissues of our body when there's in a context of an infection of tissue repair from an injury, there is a need of at least two main stages, attraction and adhesion. So one example that I like to use in my lectures about the attraction and adhesion of these immune cells to the tissues, and works very similar to what happens in a dating stage between a couple. For example, they are going to develop this relationship. So initially it's the dating stage. They wear nice clothes, they start using those nice perfumes, the partner is pleasant to be with and close by. And this is kind of like contributing with the flirting and the attraction between the couple. And then afterwards, they start developing. They talk about things that are of mutual interest. There is affinity, there is connection in this mind connection or brainstorm that they are developing afterwards. So initially there is the attraction stage, and afterwards they attach. There is affinity, there is adhesion. The same happens with our immunity, right? And I like to emphasize that it's not in every place of our body that it will happen, the trafficking of the leukocytes. So if you think about the trafficking of these leukocytes, it always happens from the bloodstream to the tissue, right? So the infected tissue or the injured tissue. But it will not come from any blood vessel. We have several different types and size of blood vessels in our body. If you think about the high caliber blood vessels, for example, the arteries and the veins. And then connected to the arteries, we have the arterioles. And then connected to the arterioles, we have the small caliber ones that don't even have elasticity. They don't dilate that efficiently, the capillaries. And then afterwards, now going towards the vein, we have the we still have the capillaries, and then we have the postcapillary venules. So they are venules connected to the veins. So these postcapillary venules, they have some plasticity to receive the blood flow there, but these are the ones actually that are mostly connected with the tissue. They will respond to the infection and they will express the ligands that will allow the leukocytes stopping there to infiltrate the sites of infection or injured tissue, right? So if we are talking about the trafficking of leukocytes, we are talking about the postcapillary venules, okay? So they're connected to the veins. So these postcapillary venules that will allow the leukocytes to attach to the endothelial cells composing the pipes that will infiltrate the tissue. So this lecture today is mostly to represent in the cellular and molecular level how this trafficking works, because my previous lecture was just an introduction, bringing the conceptions for you, but I want to show today mostly the proteins that are involved, the ligands, the cells, what's the structure of the postcapillary venules that we are talking about, what kind of tissue is infected, how happens the signaling process there, right? It's actually beautiful and very easy to understand. So looking to the schematics here, page 50 of this book, of the Cellular and Molecular Immunology from Abbas, let's look at this red area here. If you can see here, there's like actually a big cell. It's not very easy to understand that this is actually a cell, but there's the nuclei here, and then the cell membrane surrounding here. It's like a big cell. So we are looking here in this figure like the wall is the other side, is the other wall, okay? And it's imagined that this blood vessel is cut in the middle. So we can see here the blood flow. It's going from the left to the right, and imagine all the leukocytes and other blood cells that would be traveling towards the blood flow. And then we have the leukocyte, the immune cells, random immune cell here, right? So they are traveling here. All this blood vessel, this postcapillary venule is connected with the tissue, right? So could be your skin, could be the surface of or a layer of another organ, whatever. And these cells here in the bottom, they are the floor of the postcapillary venule connecting the venule with the tissue, right? They are endothelial cells. They are part of this entire wall here. But anyways, Turns out that when you have an infection, and we are talking now in this example as an intracellular infection, we have here the macrophages are engulfing here the microbes. It could be like, it looks like a virus, right? So it's kind of like digesting the virus here. It's activating a cellular signaling process inside of the macrophages. The macrophages, they are trying to resist infection here. They are resident here. They are innate immunity cells. They are already living there for a while, and the virus is trying to infect this part of the tissue. Could be a mucosa, could be the respiratory tract. And the macrophage is trying to hold on, and it will start eating this virus here. And at the same time that this fight starts, the macrophage will secrete what we call cytokines. They are pro-inflammatory cytokines. For example, the TNF or the IL-1, TNF, tumor necrosis factor, or IL-1, received this name IL, it comes from interleukin 1. So these cytokines, they are pro-inflammatory cytokines. They are secreted here inside of the tissue, while the macrophage is fighting the pathogen, this virus here, could even be COVID. And then these cytokines will start stimulating here the endothelial cells that are surrounding this postcapillary venule, right? So these endothelial cells will start producing here a substance, a molecule called selectin. So I didn't talk about the selectins in the previous lecture. It was more conceptual lecture in the previous lecture. I most emphasized that the adhesion molecules like integrins. But the selectin is also an important component of this trafficking here. And pretty much what we are seeing here is the rolling process of the leukocytes that will enable its trafficking in the tissues. But anyways, so the selectins here, they have this purple structure here. And you see that they can attach to something. So they have this open mouth here in the top. Like that, it's kind of like could have an affinity with something that is expressed where? In the leukocyte. So selectins, they are receptors. They are proteins that are meant to bind to carbohydrate structures. Carbohydrates like sugar, right? So everything that you have sugar in the leukocytes have several proteins. There are glycoproteins in the surface. Glyco comes from sugar, so carbohydrate attached to proteins. So the selectin will kind of bind to these fragments of sugar that are part of the proteins or elsewhere in the surface of the leukocytes. It's not a very strong interaction. It's actually a very weak interaction, but it's sufficient to start putting some brakes in the leukocytes, because the leukocytes are traveling very fast here in the blood flow, throwing the leukocytes from place A to B, and then the leukocytes start like attaching to these selectins here, so they will start rolling and reducing the speed. It's kind of a brake, right, in the leukocytes. So the selectins are produced in response to the TNF and IL-1 produced by macrophages that stimulate the endothelial cells here, right? And they will find these receptors, this kind of sugary like molecules expressed in the leukocytes. This will be the first break that the leukocytes will have. But they will not stop yet. They keep rolling here in a reduced speed. The macrophages that are initially producing these cytokines, they are also producing the famous chemokines, the chemoattractants, right? I mentioned in the previous lecture, the chemoattractants, they are like perfumes. So they're kind of a scent. There are different types of perfumes, and people that like these perfumes, people that like other types of perfumes. For some particular immune cells, there are specific perfumes or molecules that the macrophage will produce to recruit them. So in this case here, we have these chemokines that they will attach this substance, which is called a proteoglycan, right? So the chemokine will be here in the surface, the top surface of this proteoglycan, and that will bind to a particular receptor that is in the leukocyte. So this receptor is an adhesion protein. It's a very high affinity protein, but in the beginning is dormant. I would say in the beginning this protein is with a low affinity to its receptors, right? So it's an adhesion protein, but it's in a resting state, low affinity. Here in the schematics, you can even see like it's kind of opened. It will not bind efficiently to anything. When there's this marriage, this binding here, it tells the leukocyte, hey, it's time to activate your adhesion proteins, the real ones, the integrins, right? So they are initially in that resting state. They will now take the conformation to bind to the adapters that will be also expressed in the endothelial cells here in response to these macrophages that are activated. So now that the chemokine attached to the receptor, it tells the leukocyte, hey, let's organize these adhesion proteins in the surface to adopt the conformation that is likely to bind its ligands in the endothelial cells. And this is what happens with time. So while the leukocytes are rolling here, the chemokine is telling it to change the conformation here of the integrin. And now the adapter is in its perfect shape to have a high affinity with the ligands that are expressed in the site of inflammation. So if this has an inflammation characteristics, excuse me, turns out that these ligands will be highly expressed here, and they will attach to these integrins. And then now the leukocyte stops rolling, and it will even like be with this smashed phenotype here, and it will start now infiltrating between the endothelial cells inside of the tissue, where it finds the extracellular matrix there, and it will exert their effector functions to fight the pathogen. So now the macrophage managed to attract leukocytes coming from the blood flow to help him or her to fight this infection. It's very beautiful. So in our example here, the macrophage here is fighting, for example, a virus. So it could be very well that these chemokines here, they would be like CCL4. And CCL4 has high affinity to the receptor CCR5, which could be expressed in these leukocytes. So these leukocytes, they could be T cells, dendritic cells, monocytes, and natural killer cells. Why? T cell, dendritic cells, monocytes, and natural killer cells, they are the ones expressing the receptor for a chemokine CCL4. So the receptor is CCR5. So this chemokine CCL4 cannot bind to other receptors. It will not recruit, for example, neutrophils. It will not recruit eosinophils and so forth. No, it will recruit T cells, dendritic cells, natural killer cells, and monocytes to come here. And by the way, the CCR5 receptor, mostly expressed in the T cells, is actually the pathway of entrance of the HIV. So HIV, the virus, will usually bind to the CCR5 receptor, which is used for the CCL4 chemokine to bind and activate the infiltration of these T cells. So HIV infects usually T lymphocytes of the type CD4 cells. So CD4 T cells that express the CCR5 is one of the binding ligands of HIV. So we pretty much cover now in the cellular and molecular level the 4 major steps for the rolling of the leukocytes to infiltrate an infected tissue. First, you have the expression of the selectins in response to the cytokines that, for example, macrophages are secreting there in the affected area. Selectins will put some brakes. They bind to carbohydrates in the leukocytes. The leukocytes now will reduce the speed that they are traveling through the blood flow. They start rolling there, and then the endothelial cells start secreting in response to the infection the chemokines, these perfumes, chemoattractants. And the chemokines will bind to their receptors in the leukocytes to tell them, now you need to express the high affinity adhesion proteins. Now it's not only protein attaching to sugar or carbohydrate, now it's protein attaching to protein. It's even stronger interaction, right? And that will enable now the anchor of these leukocytes, and then the 4th stage is now the infiltration. It penetrates the tissue to exert its effector functions. So I hope that you liked this lecture, and if you have any comments or questions, please write them in the comments below. I'm more than happy to come back here and answer for you. And if you have also suggestion topics that you have in mind that would like me to come and discuss about immunology and disease in general, please write them in the comments, and I'm happy to bring a video, a short comment about that, okay? So don't forget to subscribe in the channel and share with your friends. Let's teach immunology to everyone. Bye-bye.